and 1 year in the INRAE in Lyon, the Laboratory of Plant Development and Reproduction in France. As a scientist, he has been dedicated to improve sunflower yield stability in a changing environment by genomics and genetics, and developed the high-throughput phenotyping platform ELIAPHEN at the Sunflower Genomic and the Sunflower Genomic web portal ELIAGEN. Dr. Langlade has coordinated several collaborative projects, such as the Sunflower Genome Sequencing Project and the Consortium, the investment for a future project Sunrise for the sunflower resources to improve yield stability in a changing environment, and also some European projects like Horizon LX, use of external helium to species to mitigate climate change impact and feedstock and ecosystem services provided by sunflowers. So, Dr. Langlade, Nicolas.
Thank you. Thank you for the introduction. We really want to thank the organizers for this amazing meeting. And when I was contacted by June to do this plenary session, she asked me to present an overview of our activities on high-throughput phenotyping. And bringing together all our activities was quite new for me, and so this is the first time I will present these activities with this angle. I hope this will be clear. So, I think we've seen from the resume Etienne explained that I'm very interested in genetics, and I think genetics really is linking the genotype and the phenotype. The genotype being the molecular factors that are transmitted from one generation to the other, but the phenotype needs to be acquired every time, and here is a very this is a very basics of the genetics. And during the last 2 decades, we've seen a great revolution in the genomic point of view. We've been through some singular molecular markers to high-throughput genotyping, whole genome sequencing, genome editing, etc. So nowadays we've got tens of sunflower genomes available. Of course, hundreds are sequenced, but very soon we all realize that we will have access to the genomes of all the genetic resources of sunflower for the species. In the same time, on the phenotypic point of view, this is more tricky because the phenotype needs to be acquired every time in every environment, every management, crop management protocol, etc. So we also are moving more slowly, but we are moving towards automation and high throughput, from manual phenotyping to robotized and automatic phenotyping. And here, this is very complex. The physiology of the plant needs to be described, the photosynthesis, the transpiration, and of course, the development of the plants with the architecture, its growth and senescence, up to its interaction with the biotic environment in symbiosis or pathological relationships. So, really, we are in this very exciting time where we are developing these genomic resources, and together we need to develop these phenomic resources and tools for the species. And I think now really the new frontier in genetics is really in the development of these phenomic tools, and this is very important for the community. So to tackle this challenge in France, we have developed a network called Phenomemphasis Network. Here you see the different, it's a network of labs all over France with the white dots. And you've got Paris in the north. And in Toulouse, we're really lucky because these phenomic platforms, they tackle either field phenotyping or control condition phenotyping or even molecular phenotyping. And in Toulouse, we are lucky because we have all three the different levels of phenotyping. So Toulouse is here. And when you look at the campus, the INRAE campus in the south of Toulouse, here you would have the different buildings and the different facilities. And so we have control condition facilities, TPMP here. We have the ELIAPHEN semi-controlled facilities here, and the field facility has a good phenotyping all over the labs. And what is also really an opportunity for us on the sunflower communities that we are really all concentrated in this place with Stefan Muno's team, my team on abiotic stress tolerance, the bioinformatics teams in genomics, the agronomy lab with Philippe Debac and Pierre Casadebet, the Hathaway Phenotype platform and the Genetic Resource Center for Sunflower. So it's really an opportunity to have all this sunflower expertise and the Hathaway phenotyping facilities in the same place. And here I'm going to present you a bit hints of things that are done on sunflower with different phenomic facilities. So first, and I will go very quickly on this, the control conditions facility, we've got a greenhouse that can have up to 500 plants. Of course, there are 3D RGB cameras, multispectral cameras, and fluorescence cameras. So we can describe some physiological parameters, but more classically by volume and the hence the growth and senescence of the plant. We have, and then with this facility we can set up scenarios of drought, web and biotic stress interactions. And this is contained, so we can have genome edited or genetically modified plants and pathogens, and some trials have been done organically with Stefan. But the great limit of this facility is the size of the plant that we can be on what we can have. First movie, yeah, things that we can acquire automatically here, and then from this this movie, we can get the volume of the plant, the part of the senescent, the part of the plant, and when we start the drought stress on the right at flowering, then we see that of course the plant is senescing more quickly, and this can characterize this genotype. So this is a dwarf genotype that we can work in this facility here. So if you're interested and go further and collaborate with us on sunflower, you can contact me or Peter's person in charge of this facility and this generic email address Pheno2. So I will spend more time on describing what we do on the two other facilities. So the oldest one, the first one in the place was actually the semi-controlled facility that we named EDFN. So this was created 10 years ago. And this is basically a platform, an outdoor platform surrounded by wind screens, and where a robot can manage plants. So it will manage drought scenarios specifically for each plant. I forgot to mention there is 1,300 plants that can be grown at the same time in this plant. And this robot will go through and water the plant according to the specific scenarios and acquire some images that we can phenotype the plant with. And so here, for instance, if we take an example, we have one genotype. We submit it to different drought stress levels. So here, 7 drought stress levels. That the drought is characterized with this FTSW. And what is very important is that we can do it through a very long time period. So here it's 1.5 months, starting at flowering until full maturity of the plant. And every day the robot will come, water the plant, and adjust this 1,000 kernel weight, the seed weight, or the biomass of the plant. We can quantify very precisely how this specific genotype responded to drought. And this was published in 2019. And so you understand that using this tool just only at this step to set very specific quantitative drought stress on one genotype using classical phenotyping with a yield, we are able to characterize our germplasm. And one example here is that we've done GWAS for drought tolerance. We identified some QTL, developed some neurogenic carrying either haplotype for this QTL. And we can see that the favorable and unfavorable allele will respond very differently to drought, to final drought, when you look at seed or when you look at biomass. And so you can start to understand a bit how this QTL and the genes behind work to make this plant tolerant. But of course, what is important in genetics is to discover new haplotypes, new genes, so that breeding programs can incorporate and make more tolerant varieties. So this is an example where we use wild Neanthus donors coming from different places in the US, introgress them into a cultivated background, and then test these specific QTL haplotypes using this same protocol. And you can see that the green genotypes would respond very differently from the blue one according to their origin in the US. So this way we're able to identify new regions controlling drought tolerance and also haplotypes for these QTLs. But one use of the platform that really motivated the creation of the platform was actually to be able to characterize more precisely crop parameters. So crop model parameters in sunflower were really lucky because we had this work done by Pierre Casadebeg to develop the central crop model. So classically, you would measure the water losses manually, you would measure the leaf dimensions manually. This was done like that in 2011. And then from that, you work out crop model parameters. So this is an example here. You would have the x-axis is the stress level, the y-axis is the leaf expansion high-throughput phenotyping robot. We actually acquire the images, extract features, develop a machine learning model, and then we are able to estimate automatically the leaf area and automatically the crop model parameters for the plants. And so this increase in throughput is really key because then you can address genetics. Otherwise, it's very heavy. So one use, classical use is, as I showed you earlier, is to characterize QTLs, characterizing haplotypes, screen panels, make genetic discovery, okay? But another use is, which I think is really important, is to define ideotypes. What ideotype is relevant for drought tolerance in the future? What we've done in this publication in 2019 was to define what would be favorable or unfavorable in the climate change context regarding drought, and regarding the sensitivity for leaf expansion or transpiration. What we showed that using simulation was that you would need, in the climate change context, what is favorable is to be sensitive for transpiration, sensitive to leaf expansions, meaning that you respond very early when there is a drought stress. And it would be unfavorable to be non-sensitive and response too late. But, for instance, we also tested this question of drought tolerance in changing management techniques. So if we incorporate cover crops before sunflower, this actually changes the nutritional status of the sunflower during its growth. And what we showed in this recent published work is that in this case, it is important to be sensitive only for the leaf expansion, but not necessarily for the transpiration. This is an independent process, independent physiological process, and you don't necessarily need to be sensitive for transpiration when you have these cover crops before. So this is interesting to understand a bit these ideotypes of drought tolerance in these different contexts, and then place, thanks to the throughput of the platform, we can characterize the new varieties and place them in this phenotypic space and identify if some varieties would be actually favorable in the climate change context or in changing management. So this is what we can use this platform for. We can use it in many ways, and it has been used in different publications over the years. Of course, in functional genomics, in ecophysiology and agronomy, as I mentioned, for phenomic development, and this was in collaboration with the INRA team, Sorbonne, University of Vancouver, Athens, and India. New developments on the Leafin platform are really to improve capacities to have these abiotic stress scenarios, and so we implemented a new convectors so we can have heat stress on the platform and increase the temperature by 6 to 10 degrees over the day, but over a long period. So the experiments we do are over a long period, so in 1 month or 2 months we increase the temperature to simulate the climate change. We also have combined stress scenarios with cold stress in a growth chamber and transfer of plants on the platform, or of course the drunken heat, as you can guess. So it's not only this platform we use on Sunflower and in Toulouse. We use a lot also the Phenomobile is a large robot that goes in the field, so it's a bit at the beginning, but yeah. The Phenomobile goes in the field and it will, with a head, it will scan the crop and the plots on the top, and you would see it moves above the plot and acquire images, different RGB multispectral images, or it scans with the LiDAR the plants, and you have the whole plots acquired in 3D point clouds. So this is used since some time now. Oops. Yeah, for RGB high-resolution images, of course you can do plant counting, height, measurements of height, measure the biovolume of the plants. It's mostly used on, I think it's mostly useful for complex species cover crops, but not specifically sunflower, I think. For sunflower, we use it more at this stage as a proof of concept in order to develop the pipelines that we want to upscale at the drone level. Because of course, eventually we want to work on this genetic by-environment interactions, and we need to acquire these phenotypes in many environments, as many as possible. And so these tools, like the Phenomobile, is the most interesting thing, in my opinion, is to develop it for proof of concept for very specific physiological traits. So we'll move to the UAV high-throughput phenotyping things that we do both in-house and very much in collaboration with the company Hyphen. So this become quite classical nowadays. So we can do plant counting, flowering, capsule counting, height, even if it's let's not enter into too much in details, but height, biovolumes, green covers. So these are fairly classical traits now. And what I want to present you is to how we develop more dynamic and physiological traits that are very difficult to tackle. And specifically, we wanted to understand the senescence of the plant and how this relates, of course, to the environment and the abiotic environment. And in this case, we developed with Hyphen and INRAE teams a tool to invert this ProCell model, a co-physiological model, radiation model ProCell, and estimate some F cover. So here you would have the cover of the plot that is covered by green sunflower, and different date of acquisitions. Of course, the plant grows at the beginning, there's flowering, so there's the plateau, and then the senescence. And if you normalize this plateau and we'll work on the final part of the curve, you realize that this area above the curve of the F cover is actually a proxy for leaf senescence. And so we decided to value measurements into genomic prediction measurements. And this was actually interesting to see that the genomic prediction were actually more accurate in some way, close to our proxy, to our phenomic proxy, that you can see here on the middle right. So we're happy with this proxy for leaf senescence, and we could validate some QTL and genomic prediction using this tool. So this is useful. And now we want to have this protocol for this complex trait. But in fact, this is done at the plot level. And this is very, I think, important for us, and we have the opportunity in Sunflower because it's such a large plant, to work even with UAV phenotyping at the individual plant level, because it will open doors to work on diversity within the plot and on a statistical level to remove outliers, to have better genetic values, but also to work on genetic diversity within genotypes or populations. And so you could define if your population is actually becoming inbred, or if it's more variable, genetically variable within the population, which is interesting on a genetic point of view, of course. But you could also do forward genetics and screen your plant in nurseries at the individual plant level if your pipeline is fast enough. So we wanted to go in this direction on this trait. And here, here you will have a line in your nursery. It's growing, and so you separate the different plants. You have to recognize each plant on each stage, which is the trickiest part. You separate the different plants, and then you can have this growth curve for individual plants in your nursery or in the field. And if you do this, you can calculate this proxy for leaf senescence on the right, but you can also calculate some vigor proxies in red here, which is, of course, a very interesting trait in some environments, such as cold or drought at early stages. So you have these different proxies for vigor and leaf senescence. And when you do this on a large field experiment with a population, which is a nested association lagging population, we had 574 lines across on 3 testers, so it's 1,700 plots. And we were able to phenotype leaf senescence for 140,000 individual plants. And so you can understand very clearly that from a statistical point of view, it's really interesting. And you can work out some correlations between yield, farm time, QTLs, and you have colocalization between this yield traits, this vigor traits, and leaf senescence trait, for instance. And what we want to do now is understand better how this leaf senescence relates to the yield. And that's what we are doing in this European project, LX, that started last year. We want to develop also new Phenomics tools to understand the leaf senescence, how carbon and nitrogen from the leaves are transferred to seeds to define new stages, and understand how the yield is constructed in these different environments, but using Phenomics approaches. So I told you that we had 3 platforms. That was true until 2024. Now we have a 4th one that is really rising quickly. We have a float of cameras that allow to track image trap the pollinators. So you would have images of sunflowers here during night or day. Image recognition of the pollinator or the insects on it. So here we have models now that work for moths, bumblebees, and bees. And some data here with 3 genotypes and the vegetation levels for moths and bees. And you can see there are really dramatic differences between genotypes for attractiveness. And what was quite surprising for us was the importance of moth for it. And so now I'm finishing. I want to conclude, like I said, that we are really into this field of developing the Phenomics tools for genetics, both on the genomic level since many years, but now on the Phenomics level. Here I didn't present Phenomics tools for disease screening, but we've seen that UBC, INRA, for instance, have developed some for sunflower, and this is really becoming very important and interesting. And this will be also in interaction with this leaf senescence and erotic architecture traits that I mentioned. I just want to emphasize the importance for our community to have databases and ontologies and describe properly our phenotypes and our protocols in the papers. we're really happy to help in any way for this, for the community, to either store on long term some data for in a fair way, or to help describing it in the databases in general. I want to here thank really the work done at UBC for the chromatology of sunflower, and also photos that are amazing. And here we have also our databases, of course, at INRAE. So thank you to all the people involved in this. Just to mention, one name is Nicolas LANGLADE for the ELIAFIN platform, but all the teams, we are recruiting in the coming year postdocs, researchers, and PhDs, so please contact me if you're interested in our work, and some funding. Thank you. So thank you for your attention.